Hi guys. I hope that you had a great weekend. Um, Today we're going to finish talking about antigen presentation by MHC. In the slides, I have kind of the rest of the stuff I wanted to say about antigen presentation with MHC, but then I've got a few kind of little other directions at the end of interesting things that are related to this stuff. So we'll see how many of those interesting directions I end up having the opportunity to talk about. If you recall, we talked about the MHC Class I processing and presentation pathway on Friday. And I wrote the 6 steps that we discussed, as well as how those 6 steps were achieved for MHC Class I over on this chalkboard to my right, your left. And we'll be sort of comparing and contrasting that with Class II today. So, as I just said, today we're going to be thinking about this same the same process for MHC Class II. And so I just want to remind you a few general things about MHC Class II as we go into this process. So you can remember that MHC Class II is made of both an alpha and a beta chain that come together to bind to peptide. This interaction with T cell receptor and MHC plus 2 and peptide is stabilized by the CD4 co-receptor. And so you can see CD4 here. So of course we are presenting antigen to CD4 positive T cells. And MHC class 2 is really, for our purposes, only presented by B cells, macrophages, and dendritic cells. This is something that sort of turns on and off and is not a constitutive process. One other thing that I mentioned before about MHC class 2 that is going to be pretty important today is that I told you about this complex of MHC class 2 plus peptide. Just like with class 1, we don't get the complex fully folded correctly until we have all of the partners there. But the key thing that I mentioned last time about the MHC class 2 plus peptide complex is that it is an incredibly stable molecule. It is what is known as SDS stable. So that means it's really, really, really hard to break that apart, which is going to be important in one of the steps that we see today. There's one other thing that I want to point out relevant to some things that we're going to see today. And it goes back to the HLA locus in humans, or it would be the H2 locus in mice. One thing that you can see on these images is that the MHC class 2 proteins do actually have those alpha and beta chains, and so there are actually separate alpha and beta genes for all the MHC class 2s. You can also see, you can't really see that as much in the view from the top, but you can see it on the bottom. But what I wanted you to notice in this image from your textbook, which is the image on the top, is that it shows in, for example, the class 1 region, the MHC class 1 genes that I told you about that are also listed on the bottom, A, B, and C, but it also has some other things up there, E, F, and G. Similarly, you can see with class 2, we've got DP, we've got DQ, we've got DR, but you can also see some other stuff like DM and DO listed up there. All of those additional molecules are molecules that are involved in sort of helping with some aspect of MHC presentation or are involved in some kind of weird non-classical MHC presentation. I will mention HLA-E and HLA-G way later in the semester. So when they come up again, note that they're kind of MHC-like molecules that are in this area. Today, as we think about class 2 processing, I am going to mention HLA-DM. In mice, it's called H2M because it's a key protein that helps the process of class 2 presentation. And so if you had noticed in your textbook that there were some letters I hadn't told you about in this figure, know that they're just kind of like the friends or the helpers dealing with MHC presentation. So we're thinking here about the MHC Class II process, and I've already told you that MHC Class II is going to be presenting peptides from the endocytic pathway. Endocytic pathway can also be known as the biosynthetic secretory pathway or the secretory pathway, whatever we want to call it. I don't care. That's where MHC Class II is presenting its peptide, is getting its peptides to present. So if you think back to that image of the cell that I showed you last time with sort of the blue versus the purple or the yellow regions, now we're thinking about the yellow regions, which is where MHC Class II is getting the peptides, while Class I was getting it from the blue regions, if I'm remembering the image correctly. We're going to see the same 6 steps for Class II presentation, though again, just like with Class I, where there are a couple places where I have to be like, yeah, sure, this is a step, and it's a little wonky. There are going to be a couple in Class 2 that seem a little weird too, but that's okay. It's going to make sense. So we can start with thinking about how we acquire antigen for MHC Class 2. And the answer is actually there are 2 different ways that we can acquire antigen for MHC Class 2. One of the ways that we can acquire antigen for MHC Class 2 is shown here, and that is with the process of phagocytosis. And so you can see a cell that may bind to some receptor, and we might then have that, or a microbe binding to a receptor, and we might have that microbe phagocytosed. We're going to make a little compartment of the cell membrane and eventually close off that microbe into a compartment called the phagosome, because it was an organelle, so it's ome, but it was made by phagocytosis, so it's the phagosome. Eventually, that phagosome, as you'll see in subsequent slides, might do things like fuse with lysosomes or somehow become a lysosome. In fact, often we eventually call it a phagolysosome. And then we'll kill that microbe and start to degrade that microbe there. And so you saw this process with the innate immune response earlier in the semester. Phagocytosis is one really important type of internalization, but it's in the grand scheme of cell biology, it's not the only kind of internalization that is out there. There are actually a ton of types of internalization. You can see some of them compared in this image on the left. And what I want you to notice is that if we look at sort of phagocytosis and the internalization that's going on with phagocytosis here compared to the other forms of internalization, with phagocytosis, we're pulling in way bigger stuff than we were with any of the other types of internalization. And when you actually look at what's going on with phagocytosis, the cell has to do a lot of work for phagocytosis. Like, it's got to mess up its cytoskeleton in order to actually reach out and make and an area that big to grab things. It's totally reshaping its membrane. There's a huge amount of interesting, to me, cell biology that goes on with phagocytosis that is different than all of these other kinds of internalization. So the reason why I point this out is that in your body, only 3 kinds of cells know how to do phagocytosis. Phagocytosis is like a really hard thing to do. It takes a lot of work. So every cell of your body can do these other kinds of smaller internalization, but only 3 kinds of cells of your body can do phagocytosis. So I want to tell you what those 3 cell types are, and then we're going to think a little bit about why this is an issue. So the 3 cell types in your body that can do phagocytosis are neutrophils, macrophages, and dendritic cells. And that's it. Those are your 3 phagocytes. So there are 2 things you should notice if you think about that list of 3 cells in your body that can do phagocytosis. First of all, can all 3 of them present on MHC Class II? No. Neutrophils do not present on MHC Class II. And so if you remember back to when we were talking about innate immune responses, I kind of insulted neutrophils a little bit and was saying, oh, they're kind of the phagocytes that are the dumb ones. And part of that was because they didn't make inflammatory cytokines. But the other part of that is actually because they can't present on Class II. They just Pac-Man eat stuff, but don't really do anything else with it. So that's one issue that you can see here. But what is the second issue that you can imagine with those 3 types of cells I just told you about, who are the only 3 cells in your body that can do phagocytosis, and this information about MHC Class II? Yeah, David.
B cells don't perform phagocytosis, but they do MHC Class II.
Yeah, B cells are not able to do phagocytosis. They're not one of the 3 cells that does phagocytosis, but they can present on MHC Class II. So clearly, phagocytosis can't be the only option for acquiring antigen for Class II, because B cells have the ability to do it as well. And so B cells are going to be using one of these other types of internalization, not phagocytosis. B cells instead are going to be using this type of internalization called endocytosis. And as you'll see on the next slide, they actually are going to use a specific kind of endocytosis. I'm looking at these slides right now and I'm like, I feel like these 2 slides should be in the opposite order. So I'll just do them in this opposite order. So here you can see B cells have a B cell receptor that can bind to microbes. When that receptor binds to microbes, a bunch of things happen, but one of them is that we actually internalize that receptor. We bring that receptor into the cell through a process of endocytosis. And so that means not only does the receptor get brought into the cell, but whatever had bound that receptor also gets pulled in. So we're basically pulling in our microbes. Because this is that process of internalization known as endocytosis, and in this case, that endocytosis happened because of a receptor, we refer to it as receptor mediated endocytosis. And so we can also have receptor-mediated endocytosis as a way that a cell could acquire antigen in the case of MHC Class II. And that's really often what we're seeing with B cells. And so what you can see here is that when we do receptor-mediated endocytosis, our antigen is binding to the B cell receptor, and then we're internalizing the B cell receptor and the antigen. They're eventually going to get separated, and the B cell receptor is going to get recycled back up to the surface of the cell while we do other stuff with the antigen that was bound. In the grand scheme of things, whether we have started with phagocytosis or whether we have started with receptor-mediated endocytosis, we're eventually going to end up with some stuff in a vesicle in the cell. If the vesicle happened because of phagocytosis, we usually call it a phagosome. If the vesicle happened because of endocytosis, we usually call it an endosome. And these vesicles can sort of both fuse with other vesicles, can interact with other vesicles, can mature into different types of vesicles. It's a whole big process. And eventually we sort of end up at the lysosome. And so processes that happen with an endosome and a lysosome are relatively similar. I'm not going to go through all the steps, but eventually we end up with those kind of merging and maturing to end up into a lysosome. The lysis, and that's just the way that it works in the biosynthetic secretory pathway. If you end up in a phagosome or an endosome, eventually you're going to get into the lysosome. The lysosome is one of the cell's two trash cans. It's one of the two places that the cell does a lot of degradation. And so just by being in the lysosome, those antigens are going to end up undergoing proteolysis. And so there's not really like a great way to do tagging in class 2. If anything, tagging is just like, well, you were already in the secretory pathway. So it's sort of like being in secretory pathway. Or being in the lysosome tags you for destruction. So the cell didn't actually really have to do anything active. That was almost like automatic based on the presence of the antigen. And so similarly, like with class 1, acquisition was sort of a passive thing that like the cell didn't have to do anything, and it actually had to do work to tag the thing for destruction. Here, the cell had to do work to acquire the antigen, but once it acquired it, it doesn't have to do any effort to sort of tag it for destruction. And so sort of originally, we're going to have our compartment like our endosome or our phagosome that's going to just be kind of a nice normal compartment. And eventually that compartment is going to allow for the degradation of many different compounds. And a big part of the reason for that is just that these are some of the things that are in the lysosome. So the lysosome has nucleases, proteases, glycosidases, lipases, phosphatases, sulfatases, phospholipases, basically all the enzymes to degrade stuff. This is a very acidic environment, so these all happen to be enzymes that only work really well in acid. The acid also helps with degradation in general. And so again, just like we saw with the proteasome, this is sort of like a sequestered area that can destroy stuff. And anything that is in the biosynthetic secretory pathway that ends up in the lysosome gets destroyed. So Again, proteolysis, pretty easy in the class 2 pathway. Proteolysis is just going to happen because we have everything going to the lysosome. You can notice that this word is lysosome, which is different than the word lysozyme I told you about when we talked about innate immune responses, which was an enzyme. Here we're talking about an organelle, lysosome. And if you think about it, if you think back to class 1, when we thought about MHC class 1 and delivery, we really had an issue that we had to think about. And the issue was that our MHC molecule, as a transmembrane protein, is biosynthesized in the ER as part of the secretory pathway. And our peptides, because class 1 was looking for cytoplasmic peptides, our peptides were produced in the cytoplasm. So they were in different places. They were in different regions of the cell, the cytoplasm versus the secretory pathway. And we had this problem of, okay, how are we going to get them to the same place? And that was like this big issue that we had to deal with class 1 that people had with delivery. So if you think about it, you'd be like, oh, sweet. This is going to be really easy for class 2 because MHC class 2 is a transmembrane protein. It's biosynthesized in the ER. It's biosynthesized in the secretory pathway. And our peptides are made in the secretory pathway. So, like, delivery shouldn't be a problem. This should be really easy. It turns out that delivery is a problem. But it's not a problem in the same way that you think that in the same way that it was in class 1. It's sort of a sort of flipped problem a little bit. It's actually a really big problem. I don't know why I haven't edited this better. So this is a figure from your textbook that I both love and hate. Because there is one thing that is done that is incorrect artistically, and I swore I thought I had fixed it. But I definitely have not. So this figure shows the entire class 1 pathway that we saw last time, and it shows the class 2 pathway that we're seeing right now. And so again, like, woohoo, it's really easy. Class 2 is in the secretory pathway because it's being made as a transmembrane protein. The peptides are in the secretory pathway. So they all end up together and, simple. In class 1, we had to figure out how to get them across this membrane, and that was the challenge. So here's the problem. You see this black line right here? In the cell, there is no black line. It's all, if we're in a cell, like a B cell, a macrophage, or a dendritic cell that's doing both class 1 and class 2 presentation, there's no black line down the middle between the two of them. What that means is that when these peptides are coming into the ER from TAP, they could end up on class 2. There's just peptides floating around the ER. They're supposed to be for class 1. We're putting them there for class 1. But class 2 could take them. If you got rid of this black line that your textbook felt some need to draw that I do not like. And so our problem is actually, and so let's imagine for a second that one of these class 1 peptides bound to class 2. And then class 2 started to go through this pathway to further get presented. What would happen when class 2 made it here and encountered the peptides it was supposed to be binding? Don't overthink this. Wouldn't bind. Why wouldn't it bind? because it's already full. It's already bound to something. Do you think it's easy to deal with the fact that it's already full and already bound to something? No, why not? Yeah.
Incredibly stable.
It's a super stable structure. Once you get a peptide in there, it's crazy stable. It's not something that can just come out really easily. And so the key thing is you got to keep this open. You got to keep peptides out until you get that MHC to the location where it's supposed to pick up the peptides. So here the delivery problem isn't how do you get peptides to the MHC? Here the delivery problem is how do you get the open MHC to the peptides? You can even see that I wrote that as the title. So if you were going to be in charge of designing all of this, what strategies might you imagine that would help you make sure that this MHC class II molecule does not have a peptide in its binding cleft when it gets to the peptides? Can you come up with anything you might do? Yeah, Brian. So you might think about having some kind of inhibitor that inhibits the peptides from being there. And that is, in fact, really what happens. The protein that works as an inhibitor does two kind of important things in this process. So this protein is called the invariant chain. And you can see the invariant chain here. And what you can see is that the invariant chain has a transmembrane tail, and then it kind of loops through the peptide binding cleft to block the peptide binding cleft, and then it also actually keeps going for a little bit, so that you can see our whole invariant chain shown here. So one of the things that the invariant chain is doing is blocking that peptide binding cleft. It's actually sitting in the peptide binding cleft so that peptides can't get in there. And that's one of its sort of useful features. The other really useful feature is that it has this tail that actually goes into the cytoplasm. And that tail actually directs the MHC class II peptide through the secretory pathway to bring it to the right place. And so that tail is sort of like an address label that makes sure that the MHC class II ends up getting delivered to this location in the cell, to this compartment. So we have both that tail part of invariant chain that is going to help direct the traffic of MHC class II, and we have this part that is actually going through the peptide binding cleft. And in both of these images, you can see that the part of invariant chain that goes through the peptide binding cleft is shown as sort of having a unique color. So you can see here it's shown in red on the right, on the left it's shown in purple. And that's because that part of the invariant chain has a specific name. So that part of the invariant chain that's in the peptide binding cleft is known as CLIP. CLIP, class II invariant peptide. So invariant chain or CLIP, and sometimes people just talk specifically about CLIP here, is really key for delivering that MHC to the peptides so that binding can happen. And you can see this happening here. See, they didn't make a line on this one. Here's our ER. We're getting some peptides getting shot into the ER. We have them binding to class 1, and we have invariant chain and clip blocking class 2, so class 2 doesn't steal any of those peptides that wasn't supposed to. Invariant chain also helps make sure class 2 moves to the right place in the cell. And what you can notice is as we get towards the lysosome, as we get towards sort of that degradation location, all the parts of invariant chain other than clip eventually get degraded. So when we're ready to actually load those peptides onto MHC class 2, we end up just with clip left in the binding cleft. We've degraded all the rest of invariant chain. It served its purpose of helping to deliver MHC class 2. And so you can see sort of this process of MHC class 2 and clip here. But now you can also see that we're going to have a bit of another problem, or you can imagine that we're going to have a bit of a problem here. So here you can see the antigen coming in after having been phagocytosed. It's going to meet up with MHC class 2 with clip. And so if I go back to this image from your textbook or this image from your textbook, you'll realize that this is actually not drawn entirely correctly. There should be like a little green thing or something here in the peptide binding cleft, occluding or blocking the peptide binding cleft here. So what problem do we now have if we want to act? We've delivered the MHC to the peptide, but now we want the peptide to bind to the MHC molecule. What problem do we now have for binding that we're going to have to solve to make binding happen? Yeah, Brenna. The cleft is still blocked. Clip is in the way. So we want to put a peptide there, but clip is there. It's in the way. And I know this is redundant, but why is that a problem? Yeah, Jonah.
Because class is very stable.
Class 2 with a peptide in it is really stable. And so somehow we have to like get this stable complex to get rid of clip and now get one of these peptides in the peptide binding cleft. Yep.
So just for clarification, I thought class 2 was stable when it's bound to a peptide. It's still stable when it's bound to clip?
Well, clip is a peptide. Clip is a piece of a protein, right? A product that came from a protein being degraded, and so that would also be a peptide. Make sense? So we have another really cool example for how binding works. In past years, I have done a demonstration. I'm not going to do my demonstration today. Although I tell you, this demonstration, when I do it, you never forget the details of this. And in the demonstration, I'm going to tell you about a past year's demonstration instead. In the demonstration, I had a student in the class come up and help me in the front. I picked a student who was in my research lab. And the time that people still tell stories about was when I picked a student in my research lab named Tyler, who is like 6'5". So imagine he's sitting there. And I said, Tyler, you are MHC Class II. And I gave him a pen. I said, this pen is clip. And then I acted as another protein that is involved in this process. In humans, this protein is called HLA-DM. Remember I showed you HLA-DM earlier. In mice, this protein is H2M. And I did the action of HLA-DM or H2M, which is that I ran into Tyler as hard as I could. because it was in front of the class and Tyler is 6'5", for some reason I actually thought I needed to like make it really obvious. And we both had shoulders that hurt for the rest of the day. And he may now refer to it as the Marshawn Lynch incident. So you can imagine that. But the idea is actually that HLA-DM bumps into class 2 and CLIP and tries to knock the peptide out. It is actually a physical force thing. And so what you'll see happening is that HLA, here you can see MHC Class II is biosynthesized with all of invariant chain. Eventually, we degrade invariant chain and just have CLIP left in MHC Class II pocket or cleft while we're traveling. Then we get to the location of the peptides and we see MHC Class II has CLIP. It would like one of these peptides. And HLA-DM actually comes and physically interacts with MHC Class II to try to dislodge that CLIP peptide and allow other peptides to bind. And this process happens over and over again until the binding is strong enough that CLIP or that HLA-DM cannot knock the peptide off. And so really imagine there is this other protein that's coming and shoving the class 2, hoping to make it drop its pen or drop its peptide. And then other peptides come in and bind, and then it sort of tries again to see if it can knock the peptide off. And this happens iteratively until DM can no longer knock the peptide out. And so again, that's part of how we get especially strong binding at the end. Once that peptide can no longer be knocked out, then MHC class 2 is able to leave the cell. And so our binding event happens because of the action of either HLA-DM in human or H2M in mouse, aka the Marshawn Lynch incident.
So once the peptide doesn't come out, that's because it's the right peptide.
That's because the binding strength is strong enough. It can't come out. It's the right peptide. We're ready to move on to the next step.
I felt like the binding class less.
It's less specific. It's not entirely non-specific, but it's less specific. And it is less specific, not 0% specific, and also not something that we can totally predict in terms of specificity. So it looks like evolution-wise, CLIP has matched up to not. I don't think there are the only situation that I know of where CLIP ends up at the surface. So there are some situations where CLIP can end up at the surface. The only situations I know of are in mice or humans that don't have DM. Then they end up just presenting CLIP. Really, those are mice that are really messed up because their whole CD4 T cell process is messed up because they can't present on class 2 because they only present CLIP. That interesting question. Haven't thought about it. I'll see if there's anything known about that. Like I said, I know that there's a paper where somebody made the mice that didn't have H2M, and all of their class 2 got to the surface with CLIP, and so the only thing their T cells could see was CLIP. And those mice were not in great shape. But it's possible that there was some pathogen that those mice were going to be so good at defending against because they had an epitope that mimicked the CLIP epitope. But I'm not sure that we tried to infect those mice with every pathogen that ever existed. So, you know.
I was thinking more so like could a pathogen or like an antigen be knocked off? Like could it never be presented because it resembles CLIP?
Oh, like if there's some pathogen thing that maybe doesn't have a high enough affinity, possibly. I guess that's possible, but you have to remember that when we're in this situation, of having all of these peptides being degraded, we're not even degrading necessarily one protein from a bacteria. We might be degrading 500, like the bacteria might have 500 proteins, and we're degrading all of them. And so, yeah, some of the peptides might not be very good epitopes, but it's unlikely that anywhere in the entire proteome of that bacteria, you're not going to have any peptides that are going to be able to stay on. Does that make sense? Okay. Yeah.
So I was wondering for MHC class 1, does
Mhm.
that bind to the peptide in the ER while MHC class 2 does not?
Correct.
Okay, so does it bind to the peptides that are in those vesicles? Is that what?
So that's a great question. I'm trying to look at how this is set up. In putting all of this together, today I sort of edited some slides a little bit, and I actually had some slides that were specifically where does this happen, and I took those out. But I can sort of address the detail of this. And so basically what you can see here is that class 1 is going to bind its peptide in the ER. And so you can see that the binding is shown as happening in the ER. Here you can see that MHC class 2 is binding in the biosynthetic secretory pathway where the peptides were made. Like you can see the peptides here are being made in the lysosome. And you can see sort of there's another vesicle where that binding is happening. The story that I was going to mention that I will just say in like one sentence now is that people have actually tried to figure out what this vesicle is and like what the name of it is compared to some other types of vesicles we see in cell biology, and it doesn't match any of them well. And immunologists actually gave up and made their own name for the vesicle. So the place where MHC class 2 gets its peptide is in the MHC class 2 loading compartment. Because they just gave up because it doesn't actually match. Like it's not the lysosome, it's not quite an endosome, but it's a vesicle. And what vesicle it is, people can argue about.
And it could bind to that because like the MHC gets like pulled into the vesicle.
Well, so MHC is biosynthesized in the ER and goes through the secretory pathway, and it is directed to this magical location because of invariant chain. So you can see invariant chain is actually kind of helping guide. It's the address label that's getting the MHC to this point. Okay? So the one step that we haven't yet addressed for MHC class 2 is about display. For class 1, remember display was constitutive. So what that means, if you don't remember the word constitutive, is automatic. You don't have to do anything. It's another kind of passive process. The cell is just, as soon as class 1 is sort of stably holding a peptide, it goes to the surface. It's a bit different in class 2. So actually the cell that is presenting on MHC class 2 may get some peptides loaded into MHC class 2, like you can see here, but it's actually going to need a signal to put that MHC plus peptide on the cell surface. And so you need to have some kind of signal to make this happen. So this is something that is induced. So one example you can actually see here. So this is an antigen presenting cell that has not been triggered, that has not been activated or induced. And so you can see it kind of has MHC sort of scattered around the cell. Then this is what the cell looks like when it's triggered and induced. You can see now it puts all of that MHC class 2 nicely on its surface to talk to T cells. And it is a very directed process. So here you've got a T cell in these images. Here you've got an antigen presenting cell. And in green, you are actually looking at some of the places where the antigen is found. And so you can see at the beginning, this cell might interact with the T cell, and it might be like, huh, I should like show this T cell some MHC. This antigen presenting cell might get an interaction. And here you can actually see a microtubule coming out and bringing the antigen to the T cell. And so you can, and going out, touching the T cell, being like, here's your MHC that you can see. You have induced me to show you some MHC. And in fact, that microtubule can then even be retracted later on after the signaling has started. And so you can see that this is a very inducible process. In this case, this cell got this inducement because it also got some innate immune stimulation. So it's like, oh, I better be showing off my MHC. Something's going on. And so, again, the key thing you want to see about the class 2 display is that this is an inducible process. Cells are not always presenting on class 2, while they are always presenting on class 1. Marianne Bose, who is the author of the paper that I'm showing you on the right, actually came up with a name for this process. I kind of don't like the name, so that's why it's not listed here. But officially, Marianne says this is called tubulation, because it's happening on microtubules. And that's not a word. But the point of the story is that this is an inducible process, not a constitutive process. And so you can see that this is the sort of summary of these two processes that is shown in your textbook. And that's also, we have listed on the board. These processes are awesome and fascinating, but there are also some sort of unique other cool things we can think about. What I want you to remember in us thinking about MHC processing and presentation is that the whole goal here is to make a T cell response. And so what we've been looking at is what's happening in this infected cell, this virus infected cell, this macrophage, or this B cell, how we have gotten some kind of peptide and put that peptide on MHC so that that cell can talk to a T cell, a CD8 T cell if we're thinking about MHC class 1, or a CD4 T cell if we're thinking about MHC class 2. So the whole point of this discussion has been thinking about this as the antigen that binds to T cells. And this is getting us ready to kind of think about T cells more starting on Wednesday. But there are kind of, like I said, some other unique things that we can think about. One other thing that I forgot to mention to you on the summary slide is if you look at the slides I had posted for Friday, at the end, there were two other summary slides that were sort of the summary of this process from different textbooks. And they were little tables comparing it. I point those two tables out because when you're studying this material, you may find those tables useful. I'm not sure it's useful for me to read those tables to you when I've already just told you all the stuff, but that looking at that in table form might be useful for you, and it's the final two slides from Friday's slide set. That was the other thing I was going to say. Yep, Josh. Great question. So it's really about the location of the antigen. It's really about, is that antigen an antigen that comes in through phagocytosis, or is it an antigen that's in the cytoplasm? In general, viruses will be in the cytoplasm. And so by and large, viruses are going to get presented on class 2, or on class 1, I'm sorry. Oftentimes, bacteria are either phagocytosed or are living in a vesicle, and so they're going to get presented on class 2. In reality, viral antigens are also presented on class 2 through some processes that I'm not going to get to here, but viruses actually can be presented on both. Usually if I ask you about a virus on a question, I'm meaning class 1. Bacteria, we sometimes will only see on class 2. There are some bacteria that will end up in the cytoplasm. And if those bacteria end up in the cytoplasm, then they can get some of their antigens presented on class 1 as well. And to sort of segue off of that, particularly if we're thinking about class 1, that's something that was in the cytoplasm, which is often a virus. The best way to deal with a virus in particular is to kill that infected cell. So our T cell is going to be the CD8 T cell, which is going to be the killer T cell. If we're thinking about class 2, it could be bacteria. It could also be a variety of other types of antigens, like a viral antigen. And so there our CD4 T cell is going to produce cytokines that are going to be relevant for different kinds of microbes. And we'll get more into sort of which cytokines and why later on. Does that answer your question, Josh? Okay. That also helps me segue very nicely to the next point that I was going to make, which is that, so let's imagine you were a virus. And in particular, since I know a couple of you have taken virology, I will even further specify in this case, you are a very large virus with a very large genome. How do you feel about MHC class 1 if you are a virus? Really any virus, but the first examples I'm going to talk about are all very large viruses with very large genomes. How do you feel about class 1? Getting some faces. Yeah, David.
I mean, as a virus, I wouldn't be a fan of it.
You would not be a fan of it as a virus. Why?
It would present my proteins.
Mm-hmm. Yeah, it would kind of be like your big enemy, like your biggest enemy, because it's the thing that's actually like showing your presence and like foiling your Scooby-Doo plan, right? As a result, viruses and all microbes, I could do a bacteria version of this too, but I'm doing the virus version because have come up with ways that sometimes they try to avoid MHC. This honestly, I could tell you about microbes avoiding every part of the immune system we've talked about. But think this particular virus example is a key one to think about. And so I'm going to show you a few examples of some viruses and things that they have evolved to do to evade MHC class 1 recognition. Because if they just sat around and let MHC class 1 recognize them, then that would suck for them and that would be the end of that virus.
And that's because MHC class 1 is self-acquisition, when the virus infects the cell, the MHC will get.
So it's anything. So it's because class 1 is being in the cytoplasm is acquisition. So part of a virus's life is you got to be in the cytoplasm replicating. Your proteins have to be made in the cytoplasm on a ribosome. So you can't avoid being necessarily made in the cytoplasm if you're a virus. So one virus that has an interesting way of avoiding MHC class 1 is Epstein-Barr virus, or EBV. EBV is the causative agent of mono. It also causes a few types of cancers. And EBV has this protein called EBNA1. One thing I should mention about processing on class 1 and class 2, but especially in this case, class 1 in general, is that if you remember, we talked a little bit about the fact that self-peptides are always being presented, right? I told you guys last time, MHC class 1, all of your cells 24/7 are presenting whatever proteins are in them on MHC class 1. Just like T cells, am I okay? T cells, this is what I got. Am I okay? T cells, this is what I got. Am I okay? And I know that you guys were a little bit like worried about that in terms of autoimmunity issues because you're presenting all your self-peptides. And I said that's why T cell tolerance is great. One thing that does sort of play into all of this is amount of the protein that's getting made. So if you have a protein that's being made at high amount in the cell, it's going to get presented on the surface a lot, like math-wise. When viruses are infecting a cell, they tend to make huge numbers of copies of their proteins. So if your cell has a virus in it, yeah, it might pick up some self-peptides, but it's definitely picking up a lot of virus peptides because there's a lot of virus in the cell. In the case of Epstein-Barr infection, this one protein that is involved in Epstein-Barr infection, EBNA1, is made at huge levels. The virus needs so much EBNA1 in order to replicate. And so you can imagine EBNA1 is probably going to get degraded and presented on class 1 a lot and be a great target on class 1, right? But it's not, because Epstein-Barr virus has evolved in an interesting way. EBNA1, it turns out, has strings of amino acids, particularly it's these strings of glycines and alanines, that the proteasome doesn't like. And so EBNA1 can't actually get degraded in the proteasome. While most other proteins can, EBNA1 might get ubiquitin on it, but it's just going to be chilling out here outside the proteasome and not get degraded. As a result, you can imagine that Epstein-Barr infected cell is going to keep on proteolyzing other stuff, like its self-peptides. It's going to keep moving that using TAP to move them into the ER. It's going to keep presenting the self-peptides, but it's not going to present EBNA1. And so it's sort of like the virus has figured out a way to hide from T cells and from T cell killing. It's pretty clever. Some other really famous examples of viruses evading MHC Class I are done by two viruses that are shown here. They're both types of herpes viruses. HCMV has a protein called US6. Herpes simplex virus, HSV, has a protein called ICP47. No, I'm never going to ask you, tell me about, name check these proteins, but understanding the process of, how they're evading is important and cool. Both of these herpes viruses make proteins, and if you actually look at the crystal structure of these proteins, particularly ICP47, it looks structurally like a stopper. And in both cases, the protein plugs TAP. You can see in one situation it plugs it from one side. One situation it plugs it from the other side. Whatever. In either case, we're plugging TAP. Now, class 1 can be hanging out in the ER, ready to go, ready to catch its peptides. But no peptides get shot into the ER because TAP is blocked. Our transporter is blocked. And so, is MHC class 1 ever going to fold correctly? No, because it's never going to be able to get a peptide. Is it ever going to be able to go to the surface of the cell? No. So this cell is just going to stop presenting on MHC class 1 altogether. It's actually not going to present any of these herpes virus peptides. It's not going to present any self-peptides either. It's just going to stop class 1 presentation because we have basically put a block in between proteolysis and delivery in the class 2 or in the class 1 processing process. In fact, trying to figure out what these proteins did was how people found TAP. Is they're like, what does this weird virus protein bind to? And then they're like, oh, wait, TAP. Oh, that answers all of our questions about how MHC class 1 worked. Whoops. To give you another fun example, there is a virus called adenovirus E19. Adenovirus E19 makes a protein that binds to class 1 and separates it from its chaperone. And so now the peptides can come into the ER, but MHC class 1 isn't nearby. And so MHC class 1 isn't necessarily going to get those peptides as well. And so again, we're going to at least decrease class 1 presentation. We may not stop it altogether, but we're going to make it less likely to happen because we have put a block in the middle of delivery and binding in the class 1 process. as a general overview, these are a whole bunch of different viruses that all are impacting MHC class I processing and presentation. One of the ones that is really fun that I do want to mention is there's this one KSHV. It's another herpes virus protein. It actually binds to class I and pulls it off the membrane and sends it to the lysosome. There are actually versions that pull it from the Golgi. There are ones that actually pull it from the ER from different viruses. But they basically just take MHC class I and throw it in the trash can and keep pulling it off the surface of the cell. They don't like, and it's not, they're going to pull off the MHC class Is that bind their peptide. They're pulling off all of them. So again, no self-peptide gets presented in this case. We're, if, basically just going at the end and being like, nope, no MHC class I for you. My cell, I'm going to hide by just getting rid of all the MHC class Is from the cell. And these processes are all kind of related to sort of this other piece related to MHC class I, which, as I told you about, it was that self-peptides are always being presented 24/7. If self-peptides are not being presented on MHC class I, that means there is something wrong with that cell. Most famously, that means that cell is infected with one of those herpes viruses. Something is going really wrong if a cell is not doing MHC class I processing and presentation. And so we actually have an immune response to cells that are missing MHC. So we are actually checking, is this cell presenting on MHC class I 24/7? If it ever stops, we're going to kill it because there's something wrong with that cell. That is going to involve another cell type called an NK cell, or a natural killer cell, that we will see later in the semester. But what I want you to notice here is how important that self-peptide presentation is, because it is actually an indicator of cell health, that the cell is often showing its self-peptides or its whatever peptides to T cells. So I want to mention one other thing super briefly. So you guys know about, I've thought a little bit about viral escape from adaptive immune responses, and there's strong selective pressures for viruses to escape from adaptive immune responses. And we can think about this happening with antibodies. So we might imagine here's a viral protein, binds to an antibody. The virus can make a mutation so that the protein changes right at that epitope, and now the antibody no longer binds. Okay? And we might think about this a lot with, say, SARS-CoV-2. This is the story with variants. This is why we spend a lot of time thinking about variants. But I want us to think about an important thing. So let's imagine that this mutation happens in SARS-CoV-2 virus in David. So the virus is reproducing in David. The virus makes this change. Here you can see kind of a big macro version. Here's the micro version. And then David comes to my office to talk about his thesis and spreads that mutant virus to me. Is that virus going to evade my antibody responses? Especially if I got a vaccine against the original virus. Why? So yes, there likely is another epitope. Is it going to evade that epitope? Let's imagine this is the only epitope. Obviously, you are all entirely correct. There are more epitopes, and I'm not actually worried about this in real life. But if this was somehow the only epitope, would it evade antibody responses in me? Yeah, because the epitope in me is the same as the epitope in David. It's just like a part of this protein, right? what the paper that I had wanted to talk about today, that I'm not going to get a chance to talk about today, talks a little bit about some of the same thing with T cells. So now let's imagine same thing. This virus undergoes some mutations to avoid a T cell response in David. And so this is a different example of viral evasion. This is something that might happen with any virus, whether it's a big one or a small one. Can that virus pick up some point mutations and avoid T cells in David? So yeah, potentially it can. It might get some point mutations, particularly in maybe an anchor residue, so that it can no longer be presented by David's class 1 MHC. So you can imagine a virus adapting to David's type 1 MHCs and becoming invisible to CD8 T cells because that virus changed an anchor residue in one of its proteins so it can no longer be presented on David's MHC. This could be especially a problem if David was an inbred mouse who did not have a lot of diversity in his MHC. So that could happen. We see there are lots of well-known situations where we've seen that happen with certain viruses. Now let's imagine that David comes to my office to talk to me about his thesis. Is that virus going to evade my CD8 T cells? I see a couple head shakes. You're shaking your head no. Why not?
I mean, unless we have the same MHCs, which is highly
right. If the virus mutates and sort of becomes perfect for avoiding David's MHC, I got different MHC. The epitopes in me are different than the epitopes in David because my MHC is showing off different peptides. In the antibody case, the epitope is the same because it's just a free part of the protein. This is my epitope and this is David's epitope. In the case of MHC presentation, we each have different epitopes. And so if a virus tries to mutate and avoid one person's T cells, that sucks for that one person. But then if it transmits to somebody else, it's no longer has a selective advantage. It's no longer a good virus to avoid that person's immune responses. And so while we see things like viruses doing mutation, like point mutations, to evade antibodies, we really don't see the same kind of evasion of MHC. And it's because we as a population have such diverse MHC. And so like I can see all sorts of evidence in a lot of the big SARS-CoV-2 variants of look at these mutations at these epitopes that are avoiding antibodies. I can say, oh, I need to worry about my antibody response that I got from my vaccine, whether it's the original one or a new one for a new variant. My T cells don't care. My T cells are going to present whatever works best for me. And because it's just not advantageous for the virus to mutate around my MHC compared to your MHC, it's only going to help it in me. That's not going to help it worldwide. And so we also don't see kind of the same types of mutation unless we're in really unique situations like the situation in the paper that we were going to talk about. I'm going to start talking about T cell responses, particularly the T cell receptor. And so we'll be thinking about kind of VDJ and T cells on Wednesday. If you look at the practice, like old exams, and you see anything about cross-presentation or CD1, I did not talk about them this semester, so you do not need to know about cross-presentation or CD1. And remember that the problem set about MHC and antigen presentation is due on Friday. It's posted if you want to ask me any questions. So I will see you on Wednesday.